Good morning and welcome to the first grand rounds of the academic year. Welcome to everyone. I couldn't be more pleased to introduce our first speaker of the year. With all Big Ten rivalries aside, I'm delighted to introduce John Carethers. John is a good friend and colleague as a Big Ten chair. He grew up in Detroit and attended Wayne State University for both his undergraduate and his MD degrees. Graduating first in his class in AOA, he went off to Mass General for residency and then was recruited to the University of Michigan for his gastroenterology fellowship, postdoctoral research, and research experience until he was then recruited to the University of California at San Diego, where he spent nearly 15 years rising through the ranks from assistant professor to full professor with tenure. He was the GI fellowship director. He was the section chief at their VA, and then chief of the Division of Gastroenterology from 2004 to 2009. He also led their NIH-sponsored T32 training grant. In 2009, he was recruited back to Michigan for his current position as the Searle Professor of Internal Medicine and Chair of the Department of Medicine. He's also Professor of Human Genetics at the University of Michigan. He's really internationally known for his research in colorectal cancer, including mechanisms of tumor progression, tumor genetics, familial cancer, and polyposis syndromes, and colorectal cancer disparities. He's widely published. I always like to see how people got their start. His first article was about frostbite. His second was about MRSA. But by his third journal submission, first author on an Images in New England Journal, it was to do with extraintestinal manifestations of Crohn's disease. And from that point onward, he's focused on disease of the gastrointestinal system with over 130 peer-reviewed papers. In addition, 32 invited articles and other contributions, and 38 books and chapters in books. He's been very well supported throughout his career, currently being PI on 2 R01 awards and a UO1. He's served as an excellent academic citizen, performing grant review for the NIH, the VA, the AGA, and other organizations. He sits on 7 editorial boards and is a member of several extramural advisory boards as well. Other committee work includes work for the VA, the NIH, the AAMC, and the American Association for Cancer Research. In addition to all of this, he's an active teacher at all levels, students, residents, and fellows. Along the way, he's collected an honor or two. Importantly, a House Staff Teaching Award and a Mentoring Excellence Award back when he was at UCSD. He's a member of the American Society of Clinical Investigation, the Association of American Physicians. He's been elected to the National Academy of Medicine and the American Clinical and Climatological Association. Wayne State acknowledged him with a Distinguished Alumni Award in 2015, and just in the last 2 years, he was elected as a Fellow of the American Association for the Advancement of Sciences and the American Academy of Arts and Sciences, both really tremendous honors. He has served as visiting professor throughout the country and really throughout the world. And it was awfully generous of him to agree to come across the lake and visit with us today, providing grand rounds entitled Colorectal Cancer, the Role of Inflammation and DNA Mismatch Repair for Patient Outcomes. Please join me in welcoming Dr. John Carethers.
I almost didn't recognize that guy. My gosh, that was a wonderful introduction. And thank you for inviting me and actually having a wonderful dinner last night. I came in about an hour or two earlier before I met Rick and Betsy Trowbridge last night and got to walk around your Capitol. There's very few state capitals I've walked in. It's a beautiful Capitol. I hope all of you guys have been in there. Wonderful setup, learned a little about the Wisconsin Constitution. So it's just a wonderful trip. Again, thank you for inviting me. I have no disclosures, as you see on the slide. I have been working in the field of colorectal cancer for almost 25 years now, and so I'm just going to tell you a little bit about a connection that is developed over time between genetics and inflammation in the colon. And this has to do with the pathogenesis, actually, as well as the outcome of patients with colorectal cancer. So I'm just going to give you a little overview of inflammation for about the first 5 or 7 minutes with cancer, and then specifically talk about two genetic pathways that have strong links to inflammation and how that modifies the approach to patient care and their outcome. One is called microcellular instability. Some of you may have heard that already because that's been around for about 15, 20 years. And another one called EMAS, which is probably not as familiar, and that's been actually evaluated just over the last 5 or 6 years. So just an overview of inflammation. So I'm a gastroenterologist, as you heard. And almost every disease in the GI tract, every site in the GI tract has an inflammation-related neoplastic event. In the esophagus, you can get acid reflux, and it raises your risk greatly for Barrett's esophagus and esophageal adenocarcinoma. In the stomach, the World Health Organization has classified H. pylori as a class 1 carcinogen. It causes chronic gastritis, and you can get gastric adenocarcinoma as well as meltomas from that disease. Of course, any disease that leads to cirrhosis can raise your risk for liver cancer, but in particular viral hepatitis can do that, and that's been all the rage. And with all the wonderful treatments now, we have hepatitis C. Hopefully those rates will go down. Chronic pancreatitis, repeated bouts of pancreatitis, raises your risk for pancreatic cancer. And of course, inflammatory bowel disease is a prototypical disease that raises the risk for large bowel cancer in particular. And then even down to the anus, where HPV and its level of inflammation and its viral integration can cause anal cancer. So this is a common theme in the GI tract, and also common for things like inflammatory breast cancer, etc. Now, in the colon specifically, we know that as most tumors go through these stages we call tumor initiation and tumor progression. In the colon, we've classically studied this because we have the ability to look inside the colon and look at various stages of neoplasia as things potentially develop. And now, as you know, we remove polyps ad nauseum these days. That, a colonic stem cell, I'll just call it a colonocyte here, but it's really a stem cell, eventually has to have a genetic event that initiates that first adenoma. So that's called tumor initiation. In the United States, around the world, that estimate is somewhere between 3 and 5 decades. So by the time we're age 50, about a quarter of us will have an adenoma. Now, to get it to a cancer, that takes another 1 to 2 decades. And if you look at the average age of cancer in the United States, it's around 68 to 70. So if you get your adenoma somewhere around age 50, it takes 2 decades for you to turn cancer. That'll be the natural history. And there are diseases here, genetic diseases, that can accelerate each one of those sites. FAP is a germline mutation in the APC gene. It really accentuates the tumor initial. You get lots and lots of polyps, and then it still takes 1 to 2 decades for you to get cancer. So you just get polyps at age 16, and then you get your cancers at age 40. Lynch syndrome, which I'm going to talk about a little bit more, which shows the biomarker of microcellular instability because of the effects in DNA mismatch repair, it's the opposite thing. The initiation is still normal. You get a polyp still 30s, 40s, etc., but the time for a tumor progression is markedly shortened based on the nature of that defect. And so they both can give you colon cancer, but the genetic diseases tend to give it at a different age because they accelerate either one of those steps. There's also input from inflammation, and this is what I show at the top here. We don't know all the connections between what the microbiome does and the interplay between the microbiome and the immune system. But I can tell you that in some laboratories and in preliminary studies, that this can greatly influence these steps in a normal person and may influence it in even some of these genetic diseases. So I have them as question marks there, but I'm going to tell you a little bit about, at least for some of the prototypical aspects of this, particularly Lynch syndrome. If you look at what are the risk factors for colorectal cancer, and I have on the left there what's things that are geared toward more likely development of colorectal cancer, and on the right, things that are more protective. You can argue, particularly for those that are protective, almost all those things lead toward anti-inflammatory state. Particularly aspirin and NSAIDs, that's the most obvious. But you can argue omega-3 fatty acids reduces the level of inflammation, and particularly in tissues, as well as a higher fiber, lower meat diet. So you can argue that many of the things that help slow down the initiation and progression of colon cancer are anti-inflammatory. So this is another clue that links these things. Now, more specifically, my friend Andy Chan at Mass General, he's been using the nursing health profession study as his laboratory as a health services researcher, and this is now linking the anti-inflammatories with genetics. And so in this paper that he published in 2012, let's see if I can get the laser here. This on the left is probability of death, on the y-axis, and on the y is the years. And if you have a mutant PI3 kinase, and this is important because, as you know, aspirin and NSAIDs are blockers of COX-2, but it also regulates wild-type PI3 kinase. Wild-type PI3 kinase is a pro-growth type of factor or signaling pathway. But when you have a mutant, that pathway acts incessantly. But if you can give aspirin, you can actually affect outcome. Here he shows the probability of death, and this would be secondary prevention. These are people who already had a colon cancer, and we do know that aspirin can prevent can slow down the rate for primary colon cancer by almost 50%, but also can reduce someone who had a colon cancer, reduce their rate for secondary colon cancer, and it's almost nearly the same amount. But that's accentuated by the genetics of this. So in patients who took regular aspirin, their probability of death was somewhere around 5% after their first colon cancer. And without aspirin was more than 30% if you had a mutant PI3 kinase. There was no difference if you had wild type at all. And this is cancer-specific survival, and it's similar for overall mortality. So this links some of the anti-inflammatory, or at least the inflammatory piece, with some of the genetics that's going on. And this was a wonderful paper that's been well cited. I do also want to mention that another group by a guy named Gallone has put forth this whole idea of this immunoscore. This looks at the type and density and the extent of inflammation in the colon. And in this wonderful paper published in Science in 2006 showed that the level of inflammation was a better predictor than stage of disease. So if you look at this grouping here, he used CD3, which is a pan-T cell marker. It looks at all types of T cells. And those who had the high density either in the center tumor or at the inflammatory margin, so he has high, had much, much better survival than if you had low, low. Okay, these were stage 4. But it didn't matter what stage it was because they were all similar. But this could group them based on the density of those immune cells inside the tumor or at the invasive margin of the tumor. In particular, CD8 positive T cells, these are those cytotoxic T cells, and those containing granzyme B, which is a substance that can destroy other cells that are released by the cytotoxic T cells, was heavily favored in this model. And I'm going to come back to this because I'm going to tell you about another paper that he just published a few months ago as I talk about microsatellite unstable cancers. One last thing on the general things about inflammation. One of the most potent pro-inflammatory cytokines is IL-6. And you will see this cytokine, if you take the serum of a patient with cancer, it's raised in almost all cancers. It's upregulated in cancers. Upregulated not only at the tumor, but upregulated in the serum of a whole host of cancers. IL-6 is a great pro-inflammatory cytokine that promotes healing. You'll see it elevated in inflammatory bowel disease, etc. And it's produced largely by intraepithelial lymphocytes in the intestine. However, epithelial cells and tumors can also produce it as well. And I'll show you when I talk about EMAS how this is an evasive mechanism to cause more genetic damage. blockage of IL-6, you know about tocilizumab that our rheumatologists use to slow down RA, etc., although there's a side effect that you can get intestinal perforation. And if you block the driver of IL-6, IL-17A, you can actually enhance the therapeutic response in mice. So it tells you that IL-6 could be a bad player for neoplasia, and by blocking it, it might improve the outcome. So let me now switch gears. I just want to give you that background because I'm going to touch base on those as I talk about these two different pathways. So as you know, that colorectal neoplasia usually starts from a normal colonic stem cell, and it can grow through a series of histologic and gross progression till you get a larger adenoma. And if that adenoma, in some adenomas, if they're left unchecked, can acquire malignant behavior. And this is just a variety of endoscopic pictures that shows these things can look largely different. This is a adenomatous polyp, which looks typical. This is an adenomatous polyp, believe it or not, looking like this. Often you'll see exophytic with stalk, but you can get flat polyps, and if you biopsy this turned out to be a cancer. This is turned out to be a serrated adenoma that's turned into cancer, and this is also a cancer as well. So you can have a wide variety of looks, but in general it follows this particular concept for the most part with that gross progression. Now it turns out if you sample cancer, and this is from the Cancer Genome Atlas Network, they have been putting out the genetic information for a variety of cancers and publishing in these high-impact journals. This graph summarizes about 30 years of genetic information known about the colon. And the key aspect is, so I'm going to just walk through this upper graph first. So this is looking at the number of mutations accumulated in the colon cancer. And you can immediately see, but if you group it from highest number of somatic mutations in the cancer to lowest, you can easily break out two groups. And I have to point to you that this is a logarithmic scale. There are a series of tumors that have hundreds to approaching thousands of mutations in the tumor. Okay? And there's another group of tumors, which is a larger group, and it turns out this is about 15%, and this is about 85%. These 85% of tumors have mutations that are in order of maybe 5 to 10 on average mutations in the tumor. So this group is we call hypermutated, and these groups we call non-hypermutated, based on the number of somatic mutations accumulated in the tumor. When you do that grouping, you can look at the x-axis and see what type of tumors they are. It turns out that essentially all microcell unstable tumors are hypermutated. Okay, and again, as I walk through microcell instability, it's because of the nature of the defect in DNA mismatch repair. It turns out for sporadic colon cancers, the way you inactivate mismatch repair is to silence one of the mismatch repair genes called MLH1. And you can see the grouping, except for maybe two, MLH1 silencing, and this is by hypermethylation of the promoter of this gene, is essentially a match for the MSI status, because these are all sporadic tumors. And then you'll see another grouping here called CIMP. That's called CPG island methylator phenotype. Don't worry about that name now, but it's a process in which a lot of methylation occurs within the tumor. And you can see for the most part that group's here, and it's because one of its targets is MLH1 for silencing. There's a handful that don't perfectly group. Okay, so suffice to say that MSI tumors are hypermutated. This group of tumors is blown up here in this little inset. It's the exact same order, and you can see MLH1 with these gray boxes. These are hypermethylated. There's another gene here called poly. This is the polymerase epsilon. It's a DNA polymerase. And remember, DNA polymerase is what synthesizes your DNA. Turns out some mutations in polymerase there, because it can't synthesize DNA properly, also gives you a hypermutated tumor. I'm not going to really talk about that group because I'm going to focus on the mismatch repair genes. The other part of this is these two graphs down here. looking at the profile of somatic mutations within a tumor is markedly different between a hypermutated and a mutated tumor. For the most part, hypermutated tumors have a series of genes in there that have coding microsatellites. I'm going to walk you through that too. Just remember, the structure of the gene, for the most part, dictates when they get mutated. That is not the case for non-hypermutated tumors. This follows what I call the Fearon-Vogelstein model, in which you have APC, KRAS, P53 type mutation, and those are the prominent genes that drive that type of tumor. So the drivers of the tumor, the type of mutations, is markedly different between hypermutated and non-hypermutated tumors. This is shown in this depiction. I put this in a review article that Barbara Young and I wrote last year, just breaking down the three major pathways that I showed you on that slide. Chromosome instability, okay, that's mostly aneuploid tumors. Going through this, each one of these depict going from a normal colonic stem cell all the way to cancer and metastatic cancer. And this is the Fearon-Vogelstein model where you get APC involvement, mutation, KRAS activation, P53, PI3 kinase that leads to cancer. The microsatellite instability pathway, same type of histology, but instead of accumulating chromosome breaks in aneuploidy, you accumulate frame shift mutations based on the structure of those genes. The CPGI and methylated phenotype overlaps with this because one of the targets is typically MLH1 for methylation, but this is the predominant pathway seen in serrated adenomas, which tend to be more right-sided and turn into cancer. And that pathway accumulates mostly methylated genes. So what looks like colon cancer, when you look at it grossly, or grossly at least, it may have taken a different highway, or i.e. a different genetic pathway to get there. And their behavior is different. So just focusing on the microsatellite instability pathway, there are actually 5 diseases, and one of them at least will show up on your boards, that associate with defective mismatch repair. The most common is something called Lynch syndrome. You may have heard it before as hereditary non-polyposis colon cancer, but we call it Lynch syndrome because we know the genetic defect. So Lynch syndrome is a inheritance, a germline mutation of one allele, it's an autosomal dominant passage, of a defect in one of the DNA mismatch repair genes. MSH2, MLH1, MSH6, PMS2, etc. So you have one inherited germline mutation and one normal allele, okay? And remember I showed you that first slide, the tumor initiation is normal, but tumor progression is very rapid, and that's because these cause a lot of frame shift mutations that speed up that step. This is common. 3% of all colorectal cancers that you see are Lynch syndrome. The problem is many people don't identify it because not all of us don't do probably the best family history, but sometimes that can even burn you. The average age of tumor is in the 40s for this syndrome. They get colorectal cancer and other GI tract tumors, including the biliary tract. They get tumors of the female reproductive tract in women, and also the urinary tract. There's a subtype called Lee-Fermini syndrome, which is the syndrome. It's a germline mutation in these genes, but you can get certain skin lesions. And there's about a 100-fold increase over the general population, which is still a fraction of a percent, that can get glioblastomas. Basically, a good family history. If you have a high suspect for this, germline testing after genetic counseling, and then you got to carefully survey those patients. And I always tell patients that there's no reason why you can't live till 70, 80, or 90 with really good surveillance, because you want to find any early formation of neoplasia. I will point out that this genetic syndrome, Lynch syndrome, lacks a mutation in BRAF, whereas the sporadic version has mutations in BRAF. So of these 5 syndromes, this is the most common amongst germline. There's another inherited form called constitutional mismatch repair deficiency. This is principally 2 genes because they're phenotype, based on their phenotype from those 2 genes, they present later, so often families don't know that they have mixed. And instead of one germline mutation followed by somatic inactivation of the other allele, This is inherited of 2 bad genes, one from each parent. Okay, but this is extremely rare, fortunately, but you get colorectal cancers under the age of 10. So most of you guys won't see this. Again, this is a fairly rare. There are 3 somatic conditions. So I just told you about 2 inherited conditions. There are 3 somatic. The most common is what we call sporadic microsatellite unstable cancers. Of all the cancers in the United States, there's 150,000. 15% of them will have this. This is biallelic methylation of MLH1. So it's just like I showed you in that Cancer Genome Atlas. It's the methylation of one mismatch repair gene that takes this. This is common, 15% of all colon cancers. Patients tend to present slightly older. It's slightly more common in females. And you will see BRAF mutations, unlike Lynch syndrome. There's another one called Lynch-like syndrome. Now, this entity is just only been described over the past 2 years. They do have somatic mutations in mismatch repair, but nothing wrong with the germline. It's biallelic somatic mutations. So depending on if you get a germline or a somatic, you get these different syndromes. This is estimated, and this is my own estimate, this is probably about 1% of all cancers. Patients do present slightly younger, so there's probably something that we haven't discovered yet, why 2 somatic mutations would present at a younger age than biallelic methylation. But the average age is around 54 instead of closer to 70. And at present, we surveyed patients like this with Lynch. There's another one called EMAS, and I'm going to tell you about this in a more specific way in detail. So 5 different conditions that you can see with defective mismatch repair. DNA mismatch repair was recognized last year with the Nobel Prize. And this is a work of 3, there's different types of DNA repair, basic excision repair, nucleotide excision repair, and DNA mismatch repair that were recognized with the Nobel Prize. In fact, Paul Modrich, who's at Duke, did work on bacteria and really showed how the system worked in bacteria. This has now been replicating yeast, and then I would say over the last 10 to 15 years, we've understood how it works in humans. But it was just recognized. So how does mismatch repair work? Well, I mentioned these names, MSH2, MLH1. They're actually named for the bacterial proteins. But they have to work together. And this is important because at the University of Michigan, our hospital, every person who walks in with colon cancer actually gets immunohistochemistry for these proteins now. This is a standard protocol, every single one, because you can pick up Lynch syndrome. The problem with Lynch syndrome is that you don't really have any outward phenotype until they get a cancer. So if someone walks into cancer, and even if they're age 50 or 60, we'll still screen this with immunohistochemistry. These proteins have to work together. The 2 main proteins are MSH2 and MLH1. They form partners with the other proteins. So MSH2 will form with MSH6, and MSH2 will form with MSH3. And MLH1 in humans forms with PMS2. There are some other mismatch repair proteins that are involved with meiosis, but these are the principal pairings. Okay? If you lose one partner, it causes instability of the other partner. In other words, it needs the partner to be unstable. And this is shown in this IHC. So this is a staining of a patient with colon cancer who lost MLH1. MLH1 is missing. You can see MSH2 stains in a tumor, MSH6 stains in a tumor, MLH1 is gone. Because MLH1 is gone, its partner, PMS2, is also gone. If you only saw PMS2 gone, then it's a PMS2 issue. Because MLH1 still can partner with other things. So the 2, and in fact, in Lynch syndrome, the most common mutations you'll find are MLH1 and MSH2 because it takes out all of mismatch repair. When you take out these proteins, you get something called microsatellite stability, which is a biomarker, and I'll show you it's because of these frame shift mutations. And they're tumor suppressor genes, and I mentioned about the Nobel Prize. So the work in bacteria and yeast, just to give you how this gives you a feel how this really works. This works as a clamp that surveys DNA. Do you remember you have your DNA is double-stranded, and when you replicate DNA, you have a template strand and a newly synthesized strand. Remember those days? And when that happens, you have to, with expert fidelity, copy that newly synthesized strand based on that template perfectly. And you have 3 billion base pairs. On average, your polymerase that makes a mistake 1 in 10 to the 10. That's about 3 or 4 cell cycles. Okay, so it's a very rare mistake. But in here, it's drawn here, C normally pairs with G. Let's say your polymerase makes a mistake and accidentally puts a T there. These proteins, that's called a mismatch or a mismatch. So this is why it's called mismatch repair. These proteins actually form a heterodimer, as I mentioned. They actually form a clamp around DNA, and then they can find these areas of mistakes, target it, bring in the other 2 proteins, and signal and target it with an exonuclease to take out the newly synthesized strand and have the polymerase resynthesize it to make sure that it's a perfect match based on that template strand. And you can imagine if the system is inactive, you can't repair that, and you will be left with that CT mistake. And it depends on where that is. If it's in a critical gene, that gene is hosed, right? So where does microsatellite instability come from? Well, it's a little bit different because not only it recognizes single mismatches, it recognizes mistakes at repetitive sequences. These repetitive sequences are what's called microsatellites. Turns out you have 100,000 of these throughout your whole genome. And they're used for homologous recombination when the egg and sperm meet and mix your parental DNA. There's some of them within genes, coding regions. Most of these are not in coding regions. This is an example. Here's a tetranucleotide repeat, AAG. That AAG can match with any TTTC on the opposite strand. Okay? Now, think of like a broken zipper. Any one of those 8, if I got 18 of these things, let's say 17 pair with these, but one of them, because it loops out, sticks out. The next one still can pair with that TTTC. That's what we call an insertion-deletion loop, depending on is it the newly synthesized strand or the template strand. So we call it a tetranucleotide insertion-deletion loop. These proteins will also recognize these loops. And again, signal, have the exonuclease take out the DNA, and then synthesize a new strand of DNA to line these up properly. So again, if you don't have these proteins working right, the number of these will change depending on if it's the newly synthesized strand that loops out or the template strand that loops out. So you can expand or contract these. That's what we call microsatellite instability. It's the microsatellite becoming a different length. So we call that microsatellite instability. That's where that name comes from. This is shown here. So let's say I have a coding gene like TGF-beta, and it has A10 in its coding sequence, and it compares with all the 10s. This slippage happens all the time, 100,000 places in your genome, all the time. But it's reversible because as it's laying down a newly synthesized strand, it might not line properly. But if you have deficiency in mismatch repair, in this case MSH6, that slippage can become permanent, and so that essentially A10T10 becomes A9T9, and you lose one of those alleles. Likewise, the same thing happens if it's a longer microsatellite. So why did I say it's important? Because this is the profile of how these proteins work. So MSH2 will pair with 3 or 6, and their profile, based on what we call microsatellite instability high, is defined by slippage at mononucleotide and dinucleotide repeats. So we call that microsatellite instability. And that's largely the profile of if you lose PMS2, MLH1, MSH2, or only a portion of MSH3 or MS6, you get MSI high. EMAS, as you'll hear shortly, is largely an MSH3 defect. So we can kind of pair the biomarker with the genetic defect in these. So what are some common features of loss mismatch repair? So this would apply to patients with Lynch syndrome or that 15% of patients with sporadic cancer. They display microsatellite instability. That's these frame shifts. And they can lose mismatch repair protein expression, as I showed you in that one IHC. Interesting, these tumors tend to be more common on the right side. 70% of all microsatellite unstable colons are on the right side. That's the transverse colon, ascending colon, and the cecum. So if you see a right-sided tumor in a young patient, I will almost guarantee you it is a microsatellite unstable tumor. They often show poor differentiation in mucin. About 40% of these tumors are mucin. In fact, a good pathologist sometimes can look at this and look at certain features and guesstimate that that is a microsatellite unstable tumor. As I mentioned, they're hypermutated. They contain hundreds to almost 1,000 somatic mutations because of the nature of the defect in those tumors. They're actually diploid. Unlike the chromosome stability pathway where you get aneuploidy, these are almost diploid tumors because the mutations are happening at sites and frame shifts, not by chromosome breakages and loss. Now here's the other histologic feature of this. These colorectal cancers attract T cells, and that's largely because when you frame shift a developing protein, you create something called a neoantigen. So normally if you make a protein, a receptor like TGF-beta R2, you make the full length and it signals TGF-beta. But if I have an A10 in there that goes to A9, that I can only make a truncated form of that protein. That is a novel protein to the immune system. And the immune cells will react to that, and they'll aggregate in subepithelial nodules and try to contain the tumor, because this is a foreign peptide. And as I'll show you, it also makes it more susceptible to PD-1 inhibitors. It turns out that these tumors, for 5-FU, which is the standard therapy for treating colorectal cancer, these tumors are much more resistant to 5-FU. It turns out about 10% of the 5-FU gets into the DNA, which is recognized by a mismatch repair system. I won't go all into that, but they're much more resistant to 5-FU. And paradoxically, if you have a microsatellite unstable tumor with this intense immune reaction, you're likely to live longer. And it's because that intense immune reaction is much more favorable for your survival. I liken this to being the guards of a jail. And so the immune cells are the guards, and the prisoners are the tumor cells. Okay? Unfortunately, if I give 5-FU to the tumor cell, it's resistant, but your immune cells are not necessarily resistant to 5-FU. And oftentimes, if you give 5-FU to a patient with MSI-high tumor, you kill off the immune cells, and you let the prisoners out of the jail with no guards. So sometimes their survival can actually be worse. This is the histology of this. So you have poor, moderate, and well-differentiated. MSI tends to be more poorly differentiated, shows some mucinous features. This is this Crohn's-like lymphoid subepithelial. So here's the tumor, and you'll see the subepithelial lymph nodes. So these are almost like lymph nodes under the epithelium. If you see this, this is assuredly an MSI-high tumor. And that's because these cells here are reacting to those frame-shifted neoantigens. This was highlighted by a study by a guy named Schwetali. He took tumor-infiltrating lymph cells, lymphocytes from patients with MSI-high tumors. And what he did is he looked at all these different genes. So it turns out there's about 150 genes in our genome that have a coding microsatellite in them, capable of being frame-shifted and causing truncation of those. So he just created those, and he took the T cells and see if they reacted to those. And so it turns out they reacted to all of these from a patient with MSI tumors, meaning that they were already immunized against those peptides. But there was no activity from a microsatellite stable tumor. Then he also took the T cells from the peripheral blood. If you had these T cells from an MSI-high colorectal cancer patient, they reacted. A microsatellite stable, no reaction. He took a Lynch syndrome patient who had colorectal cancer, they reacted. And interestingly, he took a Lynch syndrome patient who had never had a cancer. It was shown by germline mutation, but they already had some immunoreactivity to these peptides, meaning this is probably happening on a small scale already before you even develop a tumor. Now, it's very interesting because Gallone, the guy I showed you in the science paper, has shown that if you look at, and I'm sorry this isn't favorite, but the darker colors means where the pathways are activated. And the highest pathway activated from these T cells is antigen processing and presentation of exogenous peptide antigen, which is fitting for this neoantigen theory. And if you look at the things that are activated, Th1-related genes, cytotoxic T cells, CD8 T cells, classic for reactivity. He's taken microsatellite unstable tumors, then digital pathology, look at the infiltration and type of tumors. And if you look at MSI, which is shown in the red, whether it's at the center of the tumor invasive margin, CD8 is up, CD8 is slightly up in strobe, but although it's statistically significant, intraepithelial lymphocytes are up. Granzyme B is way up, for instance, in these tumors, showing that these MSI high tumors have activation of these. And if you look at memory T cell, again, another way to show activation, the MSI high tumors are, if you look at the development of these T cells, they get activated in the microsatellite unstable tumors. So the way I think of this for MSI high tumors is that you have alteration of DNA mismatch repair. This could be a germline like Lynch or methylation MLH1 and sporadic. Then you have these frameshift mutations that could happen anywhere in the genome, but in the coding, the protein coding ones, you can get these target gene mutations like TGF-beta, activating type 2 receptor, etc. That causes this neoantigen development, infusion of T cells, induction of a T cell protective inflammation, and that helps contain the tumor. That's why these patients tend to live a little bit longer. And even though they don't respond to 5-FU, they can still live longer compared to someone who doesn't have this type of tumor. Now, Gallone went on further to show, like I said, this immunoscore. So he looked at the density of these immune cells. And the immunoscore 4, so the higher density had the better survival compared to the lower densities based on his scoring system. If you look at microsatellite stable versus microsatellite unstable tumors, 3/4 of the microsatellite unstable tumors had the highest levels of immune scores. Now, he says, from his paper, this was published in Immunity earlier this year, that these disease-specific survival as well as over-survival was more dominant with the immunoscore over the microsatellite unstable status. So here, even if a microsatellite stable tumor with a high immune score, they still had better survival or comparable survival to a microsatellite unstable high immunoscore tumor. So one other thing that this is very interesting about, and so even though they resist the 5-FU, they can be much more sensitive to checkpoint blockade. So this shows, based on his immunoscore, what is the level of PD-1 expression. As you know, PD-1, CTLA-4, these are immune checkpoints. And now they're being used, there's drugs for these, for instance, anti-PD-1 is used in melanoma treatments, etc. And it turns out the higher immune score, you had higher expression of this PD-1. And this became very important because Luis Diaz's group from Hopkins published last year in the New England Journal of Medicine showing that, looking at tumor biomarker or the size of tumor, that those that had lost mismatch repair function or were microsatellite stable responded to anti-PD-1. You can see that's in the blue and black ones. There's hardly any blue, and it grew larger. They all grew smaller. And if you were a defective mismatch repair, i.e. a microsatellite stable tumor, you had a 78% free survival compared to if you were proficient in mismatch repair, only 11%. So, and this is shown with Kaplan-Meier curves that more than double the lifespan with a PD-1 in someone who had a mismatch repair deficient tumor, progression-free or overall survival. So it turns out that resistant to 5-FU, even though you have better survival, but you can even improve the survival with a checkpoint inhibitor. So the way Gallone put this together was that if you compare microsatellite unstable versus microsatellite stable, that these get frameshift mutations, immunogenic peptides, anti-tumor T cells, increase in PD-1 expression that leads to your prolonged survival. But there could be a subset of microsatellite stable tumors that may do that as well, although he didn't fully prove that. And there may be even a subset of microsatellite unstable that don't go through this process and have a low immunoscore based on this thinking. So in this case, it's the loss of mismatch repair that leads to Neo-peptide, that leads to the immune infiltration. So now I'm going to tell you on my last piece about a completely different aspect. It involves mismatch repair, but the outcome is just the opposite. So unlike marker cell unstable tumors, which have great survival, induces this great inflammation, prolongs the patient's survival, EMAS is a consequence of inflammation and is poor as a predictor of poor survival. So what is EMAS? So EMAS is poorly named, but it stands for elevated microcellular alterations at selected tetranucleotide repeats. That's why I just use EMAS. But the key word is tetranucleotide. These are those longer repeats. And up until recently, believe it or not, even though I just showed you all this stuff about MSH3 and all this stuff, we didn't understand this fully. This is seen in a number of cancers, but it was linked to colorectal cancer first in 2008. And it turns out this is the most common mismatch repair defect in colon cancer. I mentioned classic marker sign instability is found in about 15% of all colorectal cancers. EMAS is found in 50 to 60%, and we didn't even understand it. Okay? Now some people say, well, okay, it's a biomarker, so it's tetranucleotide repeat. What the heck does that mean? So over the last several years, we figured out some of the outcome and some of the mechanisms for this. And the first clue was that it had heterogeneous expression of this one mismatch repair protein, MSH3. And just to remind you, MSH3 does these longer insertion deletion loops, unlike MSH6. And so as I showed here, think of the broken zipper model where you got this loop out of a piece of DNA. So here's an immunohistochemistry example. So here's a tumor with positive MSH3 expression. Here's a tumor with negative MSH3 expression. In fact, this tumor had lost MSH2, and so we knew it lost MSH3 because it's its partner. Now, these are tumors that showed this tetranucleotide instability. We call it EMAS. And it's very interesting. When you stain for MSH3, you get this quasi-heterogeneous expression. Grossly, so you see some cells stained, some don't. And this probably doesn't show very well in the projection here. If you look at the nuclei of these cells, you get this heterogeneous look through them. And it turns out, if you look at associations, it turns out, and this is a paper we published in 2010, about 50% of African Americans had it, but only about a quarter of white Americans had it. It was associated with advanced stage, and it was associated with peritumoral inflammation. So not the same intense inflammation you see in a classic MSI, but it looks like the inflammation was there. And so we had thought that maybe the inflammation was the cause of EMAS, but we had to prove that. If you look at outcome, and this is not from my lab, this is from a different lab from Texas, they looked at this and looked at the outcome. So remember I told you the outcome for MSI high tumors is very good, and that shows here. If you have the EMAS, it's very bad. Okay, so it's just the opposite, even though it involves a different aspect of mismatch repair. Okay, and this was as predictive as poor stage by statistics. So we saw. So when you look at this observationally, you see this inflammation around them, but we knew that these weren't causing all these frame shifts. So first we looked at, is it mutation of MSH3? Hit nowhere. Maybe it was hypermethylation of MSH3, hit nowhere. Then we looked at the expression in cells, and this is total expression. So this first line is MSH3, and we figured maybe IL-6 is a great pro-inflammatory cytokine. We looked at several cytokines, by the way, and we found no difference in the mismatch repair proteins by increasing the level of, for instance, IL-6. So we were a little bit perplexed until someone in my lab made one little observation, and we found out why. So this shows total cell. The difference is you have to look at where in the cell it happens. And this is where we hit the jackpot. So let me walk this through you. These are colon cancer cells in which MSH3 is stained with an antibody that has a green fluorescent protein on it. DAPI is just a chemical that stains nuclei, stains the genomic material. And if you look at the control, all the MSH3, which is a mismatch repair protein that repairs DNA, if you overlap that with the nucleus, you see that all the MSH3 is in the nucleus, right? If you give IL-6, so remember the previous slide showed it was the total cell. So the amount of MSH3 didn't change. But if you look at where the MSH3 is, you give IL-6, you see now this looks like this green is dispersed in the cell. You overlap with the nuclei, and it's on the cytoplasm from IL-6. And what Stephanie did in the lab is it's like taking the yolk and the egg whites separate. So she did that, separate the nucleus from the cytosol. There's ways to do that. And if you look at MSH3, you give increase in IL-6, it drops in the nucleus and picks up in the cytosol. And it's unique to MSH3. MSH6 doesn't move, MSH2 doesn't move, MSH1 doesn't move. Okay, and this changes over tenfold just by increasing IL-6. We're like, huh? You know, why would this do this? Why would a repair protein move from the nucleus to the cytosol? So we were perplexed by this. We looked at other cytokines, TNF-alpha, IL-beta, etc. None of the other ones caused the same movement at all. This was very specific to MSH3. So we said, okay, is this IL-6 we know goes down a signaling pathway that involves JAK-STAT kinase. So in most cancers, it's a trans-signaling pathway. It's a soluble IL-6 receptor. It binds to a GP130. It's at the cancer cell surface and triggers phosphorylation of JAK-STAT kinase, which then can move to the nucleus and cause transcription. And we said, okay, so we got an IL-6 antibody. We got soluble GP130 to see if we can block this interaction, and we have a drug that can block dimerization of STAT. And when we did that, you block the movement of MSH3. And without going through this in super detail, but you can see from the pictures here, his untreated IL-6, you see you now see the nice nuclei because the green is outside the nucleus. But if you use any of those blockers, all the green stays in the nucleus. Then we also put a constitutively active STAT3 to see if does it kick it out. The answer is yes. So this is the constitutively active STAT3 stained by red, the MSH3 stained by green, and so you can see the cells that got both. And all the cells that got both, it's very hard to see the nucleus because the MSH3 got dispersed, and the ones that didn't, you see nice discrete nuclei. And if you see that here's two different cell lines in which in the control, all the MSH3 is in the nucleus, not in the cytosol. And once you put the constitutively active, you start seeing it in the cytosol. So this told us that this signaling pathway was very important for this. Now, how does this apply to humans? Well, here's staining of IL-6. So we looked at about 40 different colon cancers and see is it correlated with this EMAS. The answer was absolutely yes. So those that had this EMAS positive, IL-6 was much higher, much more IL-6 was around, and those with EMAS negative, much less IL-6 was around. The other question we asked is, okay, so now we're moving this repair protein from the nucleus to the cytosol. Is that causing mutations in the nucleus? The other answer is yes. So within 2 weeks of treatment with IL-6, you start seeing. So this is a electrophoretic of a DNA microsatellite called MCYL1, and what it is, it's a tetranucleotide repeat site. And you see two peaks, one's paternal, one's maternal, and you can see the acquisition of extra ones, meaning that it frame shifted. And this is a multiple of 4 because it's a tetranucleotide, and you can see that happen at this site as well, and in a couple different cell lines. You can see this one gained some. So it turns out that within 2 weeks of IL-6, the mutation frequency at these sites was as much as 10-fold difference just with IL-6. So this tells you that the pro-inflammatory cytokine can modify mutations in the nucleus, which we didn't understand before. Now, we have some data there's a paper that we're going to submit shortly, but the bottom line is we actually did bioinformatics on MSH3 since that was the only protein that was able to move from the nucleus to the cytosol. And it turns out it had some putative nuclear localization signals on it and putative nuclear export signals on it. And by bioinformatics, and then with causing mutations in MSH3, we were able to see which ones were the bona fide ones. And it turns out there's, we call NLS1, because that was the first one we named, was the key one, and two nuclear export signals had to work together. So it turns out if you give IL-6 or inflammation, that this MSH3 can shift out of the nucleus. It turns out it's reversible. If you can block the IL-6 pathway, remove the inflammation, it can shift back in. But when it's out, you don't get repair, and you get this biomarker we call EMAS. Now, just to finish out the three somatic conditions, so EMAS is an isolated loss of function of MSH3. That's what we discovered. It's IL-6 induced, reversible, nuclear cytosolic shift. This is up to 60% more common than the frequency of classic MSI tumors. This is associated with advanced stage and disease metastasis. I show you the outcome of those patients, poor survival. There's a higher frequency in African Americans. So this, instead of classic MSI, which induces its own protective inflammation, in this case, inflammation, which we don't know what is triggered, how the inflammation is triggered, whether it's microbiome, whatever, that inflammation then triggers a defect in the mismatch repair, which is leads to a poor outcome. And so this is the way I think of this group. You have the neoplasia and tumor advancement, and then you get some type of inflammation, and I don't know the trigger for that. But once you have that inflammation, particularly the presence of IL-6, you get alteration of mismatch repair, particularly loss of function in MSH3 because it's not in its rightful place. And then things can happen. And in particular, you can cause mutations within the nucleus once it's outside the nucleus. And it turns out the biological consequence in this case is increased metastasis. And the way I put this in the whole scheme of things is that it doesn't have its own pathway, but it modifies the pathway. I think that this is agnostic to which pathway happens. Now, that's not proven, but it seems that loss of MSH3, there's no evidence that it initiates neoplasia, but certainly can modify the outcome of patients irrespective of the pathway. So there were some key questions when we observed this, and this is about to close here. How does inflammation influence genetic changes for cancer progression? And so we showed it looks like MSH3 becomes dysfunctional. How is MSH3 dysregulated in inflammation? And we show it's a cytosolic shift. So there's no mutation that happens, no germline mutation, etc. Do anti-inflammatory agents modify this? Well, that's an open question. This is the subject of my latest R01. We're going to try to answer that question. How do anti-inflammatories reduce your risk for primary and secondary colorectal cancer prevention? My guess, you can reduce the inflammation and you're reducing the cause of EMAS and these mutations that occur there. That's what I think, but that's not tested yet. What are the survival characteristics for patients whose tumors show EMAS? We know it's poor. And do patients with EMAS have a different response to chemotherapy? I'll say no, and I didn't go into this in detail, but 5-FU is reliant on the function of mismatch repair for these patients. And it turns out you only have MSH3 dysfunction, but you still have MSH6, and MSH6 does fine in recognizing the 5-FU. So that's no. Are there target genes inactivated as a result of EMAS? I went through and showed you there's target genes when you have classic MSI. Well, based on our analysis, right now, no, and I'll stop here because of the time. But however, it's associated with loss of heterozygous events, and I won't go into that in detail. But I'm going to stop there because I know there's another announcement to make.
Thank you so much, John. I had warned Dr. Carethers that we were going to just make a brief announcement, so I'll ask people to hold questions until after the session, and he'll take questions here at the podium. Thank you so much for really visiting us in a wonderful grand rounds. I'd like to just acknowledge our chief residents this morning because it's kind of a special day. Why don't the chief residents stand, please?
Let me come on this side, sorry.
Thank you. And why don't you look that way so people can see you? Emily Jocelyn, Jeff Lynn, Jess Tischendorf, and Vidya Abraham are our chief residents this year. I'll acknowledge Jess for helping put together grand rounds this year, so I appreciate that. A few years back, a number of us decided we would make a modest endowment to name our chief residents each year, and we have everybody together to make this announcement today. And what this translates to is just a little bit of extra academic money for them. Alan Bridges, if you're here, please stand. Bennett Vogelman, if Molly's here as well, please stand. Betsy Trowbridge, likewise, and myself, we put together endowments for the chief resident. So this year, Emily is the Bridges chief resident, Jeff is the Page chief resident, Jess is the Vogelman family chief resident, and Vidya is the Trowbridge chief resident. So I just want to acknowledge them and the donors who made this possible. Thank you. Great job.